Hi, everybody. So in this video, we're going to talk about the conduction system of the heart. Now, when I say the conduction system, I'm talking about the nerve fibers that are going to run through the heart that are going to tell the muscle fibers when to contract. Because we need to make sure that all of the muscle fibers contract in the correct order to make sure that we get efficient pumping of the blood through the cardiovascular system. So we need to make sure, for instance, that the atria are empty and that they've pushed all of their blood into the ventricles before the ventricles contract so that they have more blood in them so that they're more full, so we get nice full contractions, but also so that we're not pushing air, so that we're not pushing blood from the ventricles back into the atria, all of these kinds of things. So we need to make sure that all of the fibers fire in the correct order at the right time. So because of that, we have a special little system of nerves that are going to run through the heart to make sure that that happens. Now we're going to start with these first two little dots over here. These are the sinoatrial node and the atrioventricular. So sinoatrial is at the roof of the atrium. Now, this is going to be oftentimes called the pacemaker of the heart. That's because it will set a particular pace for the heart to beat. So even without nervous sensation going to it, it will still tell the heart to beat roughly 75 times a minute. Now, that may seem kind of fast, and it is. Like, that is usually far faster than most people's resting heart rates. We have to actually use nerves to tell it to slow down even. But it can set the pace all on its own. So if there is damage to a nerve, that it will still make sure that the heart continues beating. Now, whenever an impulse comes through, whenever the sinoatrial node sends an impulse out, it's going to tell the atria to contract. So the muscles of the atria are going to contract and push blood down into the ventricles. So make sure that the ventricles are filled. After the atria are finished contracting completely, that signal will now have carried down to the atrioventricular node. Now, the atrioventricular node is also another place where we have nervous innervation from the vagus nerve coming in so that we can use this to regulate heart rate as well. Now, connected to the atrioventricular nerve, we have what's called the AV bundle. So atrioventricular bundle. So it's in that space between the atrium, between the atria and the ventricles. This is also sometimes called the bundle of His, okay, H-I-I-S. I will accept either one on practicals or anything like that. Now, after we go through the AV bundle, we're going to see nervous innervation going down through our ventricular septum here, so the space in between our ventricles, and we're going to see our bundle branches here. We're going to have left and right bundle branches, and those are going to kind of wrap around the apex of the heart before they actually start innervating the individual muscle tissues themselves so that we can tell those to contract. Now, the fibers after the split around the apex of the heart, where they split out and they actually break out and travel through the myocardium, we're going to call these the Purkinje fibers. Now, these will then basically send the signal to those muscles to contract and making sure that we contract them in the correct order. Now, the easiest way to think about the order that we're contracting the heart is it's basically like we're going to contract the atria and then we're going to contract the ventricles and kind of push everything out, okay? Now, all of this is going to be controlled by our medulla oblongata. So remember that the medulla oblongata of the brainstem is what's going to actually control heart rate. So whether we need to speed it up or slow it down, we're going to have centers within the medulla oblongata that are going to take care of this. Now, they're going to figure out whether or not we need to speed up or slow down based off of different criteria. So do we need more oxygen? Well, if we will know that we need more oxygen, we'll be able to sense that using chemoreceptors in our cardiovascular system. These will be able to say, hey, there is more oxygen draw on the system than we initially thought. Say we're exercising or something like that. Or we have baroreceptors that are sensing pressure. These are going to be very important to make sure that we are moving blood through the system at a rate that is sufficient to make sure that we can get oxygen where it needs to go. Okay, now they're going to do this via both the sympathetic and parasympathetic pathways of the nervous system. So we're going to use kind of a couple of different branches here to make sure that we can regulate our cardiac activity. Now, notice that I'm not saying initiate. That means we're not sending a signal out that says, hey, beat now, beat now, beat now, beat now. Instead, it is basically going to be influencing how fast and how hard. So we can say, hey, you need to beat faster, you need to beat harder, so we can initiate earlier responses, but we're not actually saying, hey, heart, go. Now, the two types of innervation here are going to be parasympathetic and sympathetic. Parasympathetic is going to be slowing it down, okay? Now, this is going to be regulated via our vagus nerve, okay? So the right vagus nerve is going to innervate our sinoatrial node. So the right one will start at that pacemaker. The left one will go down to the AV node. Now, our sympathetic innervation is going to be the one that's going to say, hey, this is our fight or flight kind of response. So we need to speed up here. So if we say need to start moving additional oxygen tissue so that we can run away, this is going to come from our sympathetic nervous system. So remember that sympathetic is fight or flight. And if you think about being scared, you tend to think about an increasing heart rate and things like that. So this is going to be from neurons in our thoracic portion of our spinal cord, as would make sense since we're in the thoracic region. And these are going to go to our SA node, AV node, as well as some of the arteries around them. So this leads to vessel dilation. So here we can see an image of the innervation of the heart. Now in orange, we're going to have our parasympathetic innervation. So this is where we're going to have our vagus nerve, vagus meaning wander. So notice that we have our left branch going down here to talk to our atrioventricular node, and the right branch is going down here to talk to the SA node. But keep in mind that we are dealing with parasympathetic innervation. We're dealing with our medulla oblongata because of receptors that are found in all of our blood vessels. So baroreceptors sensing things like pressures, and then chemoreceptors sensing things like oxygen concentrations and stuff like that. Making sure that we can keep those at specific means. One of the ways that we can keep those things at specific means is by, well, partially controlling our respiratory rate or our heart rate. Now, keep in mind here that cardiac muscle cells have a very special shape. Remember that cardiac muscle cells oftentimes are that kind of, I'm trying to figure out how to make my fingers do this kind of a Y shape to them where they're branched, okay? That branching to the cardiac muscle cells allows multiple cardiac muscle cells to interact with one another. And this also, at those intercalated discs, allows us to send signals through those discs so that when this one goes, it can release some chemicals to initiate the action potentials in the next muscle cell. So we can continue the impulse down the line so that we can make sure that the cells all contract in the correct order. So let's go ahead and start our signal right here at our SA node. So like I said before, we send a signal out to the muscles of the atria, moving from top to bottom. It makes sense if you think about it, we're going from the top of the atria down because we're basically squeezing and squeezing and squeezing so that we can push all of that stuff out. It's kind of like squeezing a toothpaste tube. You don't usually squeeze, or at least if you want to be really efficient, you don't squeeze a toothpaste tube from the middle. You squeeze all the way at the bottom so you can squeeze all of that tissue or all of that stuff out of the bottom into or out the tube, right? So that's what we're doing here. We're going to initiate that response at the top of the atria, and we're going to move that response down towards the ventricle so that we can push all of the blood down. Now we're also going to be propagating that action potential through our cardiac muscle cells, because remember, we share sarcolemma on these things. So the muscle cells themselves are differentiated from one another via those intercalated discs, but they will have connections between their membranes so that we can send signals through those sarcoplasmic membranes and cause those muscles to go. Now, the SA node, as I mentioned before, has the ability to set the pace of your heart rate. Now, it's going to do this because it actually has some specialized slow sodium channels that are going to just slowly allow that membrane potential to kind of creep up closer and closer and closer to its threshold until bam, it goes. And then it'll go back down, and then slowly and bam, like it'll hit that threshold, then it'll initiate a response. Now, this is going to lead to heartbeats without it necessarily having to be told to initiate that response. Because these channels are nice and slow, they're going to kind of slowly go from our -60 resting membrane potential to our -40, which is our threshold. So they're going to kind of slowly allow that to change, waiting for our next go. So this is this idea of reaching that threshold. Now, once we have reached that -40 millivolt threshold, we initiate an action potential without having to receive that signal from a different nerve. At which point we're going to have our typical depolarization and repolarization happening. So we're going to depolarize, so just shoot stuff across. In this case, it's going to be calcium. It's going to be shooting across into the cell. So we're going to push lots and lots and lots of calcium into the cell, raising that membrane potential a lot. And then we're going to repolarize. So the sodium or the calcium channels close, potassium channels open so that we can push those positive ions back out and bring us back down to our resting membrane potential. And then once we're back to our resting membrane potential, we'll open it back up and do it again. Now, in general, this is going to happen around 75 beats a minute just for our SA node. However, keep in mind that we are talking to the SA node using our medulla oblongata. So your brainstem is saying, hey, we need to slow down. Like sitting right here in this chair talking to you all, even as fast as I am talking, that is not nearly enough pace for me to need 75 beats a minute. So instead, my heart rate will be slowed down using that kind of parasympathetic response from my brain. So we're getting that rest and digest kind of response. So my heart rate is going to be nice and slow so that we can keep it nice and slow so that we can keep everything nice and even. We do this using what's called our vagal tone. So this is that parasympathetic activity. It's going to kind of keep everything slow. Now, elite athletes, especially elite endurance athletes, tend to have really slow resting heart rates. Like I've seen one person with a resting heart rate in like the 40s, like mid to high 40s. He ran marathons and stuff like that. Because he ran marathons a lot, his heart was very, very strong. It worked really, really efficiently. So his resting heart rate was even lower. So here's what I was talking about before. So we have our pacemaker potential here. So this is basically those slow sodium channels open to allow sodium to kind of be creeping in and slowly raising our resting membrane potential until we hit our threshold, then bam. Now we can slow this down by putting in additional signal to keep it slow, or we can speed it up by adding in some of that sympathetic innervation to tell it to go even faster, at which point we'll send a signal and we'll reach that threshold much faster so that we can make the heart beat even more quickly. So say you're running or something like that, okay? Now, after we go through the SA node, it's gonna go all through the atria, like I said, from kind of top to bottom, and then it's gonna go down to the atrioventricular node. Now, the atrioventricular node will start a new stimulation out through the apex of the heart, okay? So these bundles are gonna go from the AV node out through the bundle of His, or the AV bundle, then that splits in our atrioventricular septa into left and right bundle branches. Those go around the apex of the heart until they eventually become our Purkinje fibers. Now it's going to be these Purkinje fibers that are going to kind of spread through the cardiac muscle fibers themselves so that we can innervate individual muscle fibers. But also keep in mind that when you start an impulse in one portion of the cardiac muscle cells, any of the cells that are around it, especially downstream, will start to react accordingly. So we don't have to tell every single cardiac muscle cell to fire. Instead, we can just tell a few, and it'll make sure that that signal gets spread out to where it will need to go. So we start at the atria, or we start at the SA node, sends a signal through the atria contract. After the atria contract, that signal has now passed down to the AV node, at which point we will send the signal down through our bundle of His, through our bundle branches, and then finally around the apex and into our Purkinje fibers. Now, once we get to the Purkinje fibers, we're going to start the contraction at the apex of the heart so we can push all of that blood out. Once again, we're squeezing from the bottom of the tube. So we're squeezing from that bottom of the toothpaste tube so we can squeeze all that blood out up through our pulmonary arteries, so our pulmonary trunk and into the pulmonary arteries, or up through the aorta. Now, one cool thing with the nerves in the after the apex of the heart, so our Purkinje fibers, is that these are actually quite large. With them being large, heavily myelinated, we can also get really, really fast action potentials moving through them so that we get this massive contraction of the basic, or essentially the entire ventricle all at the same time. I said that we start kind of at the apex, but that's really where we start the simulation. So we're going to get a little bit of the apex starting to contract first, but we really get mostly just a slap of muscle. They're all going to contract essentially at the same time because of the thickness of the diameter of these. So they can send those signals really, really fast. Imagine like using an HDMI cable versus using like a single strand of copper or something like that. It tends to be much, much faster. So this is basically our HDMI cable so that we can get really, really, really fast signal transduction out to those muscles so that essentially the muscles all contract at basically at the same time. So we get really, really smooth, but also really, really fast contraction of the heart. Now, you may have heard of someone that has a pacemaker attached to them, so an ectopic pacemaker. This is going to be one that has been added other than our SA node. Now, importantly here, if it's going to be able to regulate its own pace, we have to be able to spontaneously depolarize on their own, okay? This is usually the case if the SA node has been injured in some way. The AV node can work on its own. However, it's going to be much, much slower than the SA node. Because remember, the SA node would like things in the 75 to 100 beat kind of range. That's much, much slower than our 40 to 50 here. So If you have any questions about heart rate regulation or anything like that, please just let me know. And I guess I'll see you in the next video. Thanks. Bye.